Ladies and gentlemen, welcome. Let's dive right in. Mr. Brian Anthony from the MIT. He's the Associate Director of MIT Nano, the co-director of the Medical Electronic Device Realization Center, and the Director of the Master of Engineering in Manufacturing program at MIT. This sounds like a lot of work and like a lot of responsibility too. So I'm not sure whether you actually ever find any time to sleep at all. Maybe you're going to talk about this also. Anyway, MIT, of course, is known all around the world for its groundbreaking basic research, but not only that, but also for bringing together basic research and industry. There are maker labs there. There are spaces for bringing together startup companies and academia, and we're going to hear now about medical device innovation at MIT. Mr. Anthony, please.
So thank you for your attention. I want to thank the organizers for inviting me over. It's an opportunity to try to share a little bit about what we are doing at MIT and in the greater Boston area, and share with you the context of how we do medical device innovation, and also dig deep into some particular areas that are the focus of my group's research. As was mentioned, I'm the Associate Director of MIT Nano. That's not the building here. This building is IMES, our Institute for Medical Engineering and Science. MIT Nano, in contrast, is our new 200,000 square foot nanofabrication, nanocharacterization facility, which is at the heart of campus and really tasked with helping to improve all activities on campus, not just medical, but pharmaceuticals, biology, chemistry, computation, what have you. But I'll share today our experiences within IMES. MIT does not have a medical school, nor will we ever. IMES is an engineering-focused center, broadly organized to connect with researchers all across campus that are doing work in healthcare. We have a large group of hospitals around Boston to connect with. So what does Boston look like? As of 2014, the population of the greater Boston area, around 5 million people. Boston proper is a little bit smaller than that, 600,000, 700,000, but the greater Boston area is a little bit larger. If you look at closer to where MIT sits, MIT is the blue triangle, right along the Charles River, right across the river from Boston. Surrounding MIT, we have Tufts, Beth Israel Hospital, Mass General Hospital, Children's Hospital, to name a few. The proximity of healthcare, the proximity of all these doctors is actually pretty instrumental to our ability to be successful in developing first in human and first pilot studies in collaboration with the hospitals. My students, my postdocs, all the faculty across MIT have opportunity to walk across the river, not on the water, using the bridge, but to connect with the hospitals and to be able to rapidly get that user experience that's necessary to be impactful in healthcare. Drilling down a little bit closer, MIT are the yellow buildings. All the blue buildings are the surrounding network of companies that are either directly MedTech or are doing MedTech related activities. Since the time that I created this map, other companies have moved in. Samsung, Philips moved their North American research headquarters just down the street from MIT. Nihon Kohden opened their North American headquarters next to campus. To be adjacent not only to MIT, but to be adjacent to this innovative medical device hub. And we have the technologists at MIT, at Harvard, at Northeastern, at BU. But if we compare, say, Kendall Square, so the area of the innovation center near MIT, compare that to Silicon Valley, the capital, the professional capital, if you will, in Boston is a lot more patient, meaning they're more willing to take the time to get the return on their investment that is typically necessary to see a medical device come into market. Right? So Boston, I argue, I'm biased, is really one of the places, the premier places in the world to do medical device, to do medical instrumentation, to do AI and machine learning in medicine. So within MIT, about a decade ago, we created the Medical Electronics Device Realization Center under the recognition that the semiconductor industry has revolutionized watches, calculators, personal computers, personal communication, the proliferation of IT technologies, both computation, both the hardware and the computation, has driven down cost and improved quality throughout its history. Now, MedTech, medical devices, medical electronic devices, are far more fragmented as an industry or as a set of technology, but MIT has been able to serve as a convener to bring together the Nihon Kodens, the Philips, the GEs of the world with the analog devices, the Intels of the world, to connect the patient to devices that allow them to collect data. And from that data, we need to process it with algorithms to extract information. You don't want to give data to a doctor or to a patient. You want to give information, actionable information, and give that information to then close the loop. Right? And not losing sight of the fact that your technologies have to fit into the MedTech ecosystem. They can't break the workflow that nurses and doctors need to follow when interacting with their patients. I said this already, but sort of the vision really was to leverage the semiconductor industry and continue it on its journey of improving costs and improving quality and driving down costs the way that it has throughout its history. The model for the MedRC, and ignore the eye chart for a moment, the big points are maybe the words that have changed. So jointly, on-site, and early prototypes. So the model is most of the research activities that we do are jointly sponsored, jointly executed by clinicians, companies, and MIT. Most of the projects are industry funded. We bring in a significant amount of money from GE, from Philips, from Nihon Koden, from analog devices. We define a project that has a clinical need and a technical line of sight, a technical pathway to an actual solution. And then, quite importantly, we also bring visiting scientists from the companies to be on campus working with us. And we get the students and the postdocs that are executing on the projects into the medical environment, into the clinical environment, working with the nurses, the doctors that are giving care. Ultimately, what we're trying to do is drive down cost, improve quality, to not deal with disease by the time you're at the emergency room and your disease is rather severe, but to get that data in the context of daily living when it's more treatable, earlier, less expensive to deal with. So when I say medical electronics, when I say medical devices, that's pretty broad. That's everything from bandages to beds. Medical electronics, at least as we define it, are the big bullet areas. So wearable technologies, minimally invasive monitors, point-of-care instruments, imaging, the last centimeter problem, so data communication, and then intersections with pharma. The small topic areas are example projects where we have successfully deployed technology into the clinical environment. So this top image, for example, is behind the ear, has the form factor of a hearing aid. It was actually spun out into a startup company that measures the ballistic cardiogram, which is the pulsing of the head to the reaction force of your blood pulsing through your body. It's a good bony prominence, so even if you don't have my haircut, your skin is exposed there, and you can get sort of view of the blood flowing. You can get pulse. You can get blood oxygenation. And so a startup company run by the students that actually were doing the research is now commercializing this. The second example, there's probably 50 faculty across campus that are doing work in microfluidics, being able to develop point-of-care instrumentation to give immediate answers to what is in blood, saliva, tears, or stool. And then what I'll focus a lot on later is this bottom image. I'll skip over that for now. Just a few slides to give you the breadth of just single slides of some of these projects. This is a representative image of a microfluidic device that uses electrical signals to characterize the composition of blood. So that's a device. This is a lot of data work where it's predicting death after acute coronary syndrome by classifying and learning the information that's embedded in ECG signals. To something like this, which is an ultrasound technique, which is a potentially low-cost replacement to the hemocytometer, the techniques to count blood cells, the number of blood cells in a volume of fluid. if you are suffering from seizures, and you would typically have to wear these externally mounted seizure Holter monitors or monitors on your head. For a good class of patients, there's a need to have more less invasive or more hidden, but certainly a little bit more invasive technology, slightly implanted, so just under the skin, to get longer-term signals and longer-term understanding of what's actually happening in the brain. So my role within the Medical Electronics Device Realization Center is to identify large groupings of technology areas where we can have impact, to help bring in money from industry, to collaborate with our doctors, and to execute on these projects. My personal research group is a mix of mechanical engineers, electrical engineers, computer scientists, and we're really concerned with trying to improve the usability and diagnostic capabilities of ultrasound imaging. Ultrasound, if you're not familiar with it, has a sonographer, a doctor holding an ultrasound probe. They're interacting with the patient, looking at a sonogram, an ultrasound image, and they are implicitly controlling the acquisition state. And by acquisition state, I mean the orientation and position of the probe, the amount of pressure that's applied between the probe and the patient. Now, ultrasound, compared to other imaging modalities, is viewed as less quantitative. It's viewed as you can get a quick look and get an intuition for what's going on, but it's rather hard, actually, to get robust measurements, robust quantitative information out of the imagery. And I argue that one of the reasons for that is because of all the sources of noise, all the variability that's associated with either the patient moving or the doctor moving. I wouldn't be an engineer if I didn't have a feedback diagram in my presentation, so here's my feedback diagram. To represent the ultrasound system flow. A probe, the thing over there in red, interacting with the patient. The acquisition state is the thing that the doctor is implicitly manipulating, and they're doing some type of analysis, some type of diagnostic based on what they're seeing. Tissue dimensions, tissue shape and structure, features and property are what they're looking for. But the sources of variability, as I mentioned, is, well, how hard is the doctor pressing? What is actually the orientation and position of the probe on the body? And that's important if I want to say, for example, track a thyroid nodule and see, is it growing? If I'm even the same doctor and I acquire an image today or a week from today and a month from today and 6 months from today, and I want to track, is that thing changing? Is it growing? Are the properties changing? And if I'm not looking at exactly the same thing, how do I know? How can I actually use that sensor data to say that, oh yeah, there's something changing? And then I won't talk about the last bullet, but there's a fundamental problem, I'll call it that, in ultrasound imagers. When you're doing ultrasound, you send sound into the body, you're acting like a bat. It reflects off of boundaries, you receive sound, and the only thing you're actually measuring is time of flight, how long sound took to go in and come out. And you're converting time of flight into an image that has dimension because you've assumed a speed of sound. But the speed of sound in tissue is roughly 1,540 meters per second, but is highly variable based on the level of fat, the level of fibrosis, the patient themselves, and the tissue structure and tissue type that you're looking at. So there's a fundamental problem in the way that ultrasound systems are fundamentally constructed. We'll hold that and we'll talk about that offline. But so what do we do? Ultrasound system flow, this is what it looks like. Where we do research are in the boxes in blue. How do we add sensors to measure and control the acquisition state, to give feedback to the doctor and to give feedback to the system? So in practice, what does that look like? We have, for example, on the far left, an ultrasound probe that has been mounted to a 1 degree of freedom robot, a linear stage that we can use to control force and motion. We have, on the far right, cameras that we've mounted to the outside of the probe. And we'll talk about these in detail. But we use these add-ons, these attachments to the ultrasound probe to either measure or control the acquisition state, to give more data so that the combination of this data plus the ultrasound data allows algorithms, machine learning or AI, so human in loop or not, to get the information that we're trying to go for. Okay. So ultimately, we're trying to improve the rate, cost, and quality of imaging. So why is that important? If we look at the basidic vein, for example, on looking at the arm, and we apply now, we instrument a force, we instrument an ultrasound probe. We add the ability to just measure the amount of force that's applied during the imaging process. And we look at a vein under 1 newtons of load, 3 Newton, or 5 Newton. So certainly if you're pressing harder on the tissue, it distorts. And so if you're trying to, say, track the dimensions of an artery or a vein or of a tumor, but you're distorting it, how can you know what dimension you should really extract? Similarly, ultrasound can be used in a mode that's called elastography. Elastography is the ability to measure the tissue stiffness. So a very stiff tissue doesn't move much when you press on it. A rather soft tissue moves a lot when you press on it. The colors here correspond to different levels of elasticity. The circled regions across the quadriceps are the same patient, same location on the quadriceps, under 1, 9, and 18 newtons of load. As you can expect, as you're pressing, not on my arm, but on the leg, the harder I press, the distance between the skin and the bone and the tissue gets squished. That's the technical term, squished. But those regions are the same region, but the elasticity value is 21 kilopascals, 38 kilopascals, or 64 kilopascals. So again, what is the actual elasticity of the tissue? It's not a singular number. It actually is a curve. Its elasticity is a function of force. If you look at what doctors normally do, this is one sonographer on 3 different patients with 3 different levels of body mass index. And we see the amount of time that it takes to do a full abdominal exam, and the amount of force that's required to do that full abdominal exam is highly variable, but it's also not standard. Certainly there's reason to press harder on a patient that's a little heavier. You need to displace the tissue to get to the organs of interest. But if I wanted to then do that abdominal exam in a repeatable way later, I really currently have no way to do that. So these are 2 examples of the way that we augment a probe. A force measurement probe is on the left, a force control probe on the right. So we take a standard off-the-shelf ultrasound probe and add technology to it. So what this looks like, what we'll see in the video, is we'll see a sonographer's arm coming into the video. There's an ultrasound probe that is making contact with a phantom, a tissue mimic. And that tissue mimic is the patient. And we'll see an image. And next to the image is this force gauge. In the first section of the video, we've asked the doctor to try to do their exam, but also maintain the amount of force that they're using. And then we see that they can't do that. And then we turn the controller on, and what you'll notice, there'll be automatic motion between the patient or between the probe and tracking the patient, moving with respect to the doctor. Okay. So we first ask the doctor, try to maintain force, and they have a hard time doing it. This is mimicking the patient breathing. Things are moving all over the place. We see the image of the artery collapsing. We turn the control system on, which allows relative motion between the probe now and the doctor's hand, and we've stabilized the image. Right? So we'll see those images side by side here in a moment. Right. So when we've stabilized, eliminated either doctor motion or patient motion is on the left, and when the doctor has tried to maintain on the right. So how do we use this? So I already alluded to the fact that it's important to measure the amount of force, or if you want to get a number out of an ultrasound image, it's probably important to know how you're interacting with the patient. So we saw this quadricep data. We've done a lot of work we did a 6-year clinical trial looking at Duchenne muscular dystrophy. Duchenne muscular dystrophy affects young boys, typically fatal by the age of 25. It manifests itself by increased fat and fibrosis in the muscle tissue. The standard of care now for tracking progression and to understanding if a drug is being effective for the individual is the 6-minute walk test. The 6-minute walk test is, as it sounds, how far can the patient walk in 6 minutes? This is much better with the lighting. It's not right in my face. So as you can imagine, I'm a young boy, I'm in the doctor's office, I'm upset that I'm sick. I didn't eat that morning. So the variability associated with a 6-minute walk test is horrendous, but our goal is, well, can we develop an imaging metric? Can we use ultrasound to extract some information that allows us to replace that standard of care with the 6-minute walk test, to go from an image to relating that to something quantitative about the muscle? This is just an image of what some of the systems look like deployed in the hospital. Again, the ultrasound probe attached to a cart, some close-ups of it. The type of data that we acquire on different muscles, this is on the biceps, the quadriceps, the glutes, we collect now imagery as a function of force. Instead of saying we just want a single force, we want imagery at 1 Newton, 1.1 Newton, 1.2 Newtons, 1.3, the entire range. And once we do that in a controlled way, now we have the information that allows us to extract something interesting. So in the various work that we've done, we've been able to highlight the fact that we've been able to identify what muscles are the best muscles to monitor, to track, and what is the impact of having force. The short answer is, if you don't have force, your ability to say, is the disease progressing, is dramatically reduced. So it's extremely important to have not just the imagery, but to know how that data is actually acquired. And so these are just different metrics that have allowed us to either look at the texture of images or the elasticity in images, and there's a number of papers here that we can point to, but to try to synthesize what's the punchline of what we've been able to do. We start with an image of a muscle. We extract some numbers from that muscle. If we plot for a population, so roughly 40 healthy patients and 40 Duchenne patients, and track those numbers for, this here is texture, could be elasticity, could be anything, as a function of age, what you'd like to see is for the healthy patients, the blue dots to be on one side, and the sick patients, the DMD, to be on the other side. You'd like to say, well, there's some classifier that allows us to say sick, not sick. But I challenge you to find any classifier that separates those dots. This is when you don't know how the data is acquired. If you know how the data is acquired, you're starting to see, well, if I incorporate a modification to that metric where I've measured force, I see the blue and the red dots start to, there's a line that roughly separates those two, which is promising, but that's not what you want to do. You don't want to just classify. You want to be able to track progression. And the exciting thing about this population-based data is if you look at this metric, as a function of age is going up the vertical axis, we're seeing these numbers change, which gives us and gave us hope that, well, maybe there's a signal, once we've controlled how we acquire the data, that allows us to track progression. And indeed, that's what we've been able to find. That now this is for a single patient that we tracked over 3 years, plotting their force-controlled measure of elasticity, and comparing that to the gold standard, which is the 6-minute walk test, we're able to strongly correlate with that progression of disease. So the punchline here, measuring force is highly important. Another example, thyroid disease. Elastograms for the thyroid under different loads. If we plot those measures of elasticity for 25 different patients, what we see is the elasticity, the stiffness of the tissue, changes depending on how hard you're pressing. As it turns out, force variation that we've characterized between 3 and 5 newtons is extremely common from what a doctor will normally do. They don't know normally how hard they're pressing on tissue. But the force can vary anywhere from 2 newtons to 15 newtons. But if we say it's easy to have 3 newtons of variation, that 3 newtons of variation on average will introduce about 5 kilopascals of error or of difference in the elasticity. That 5 kilopascals is actually the difference between a healthy thyroid and a non-healthy thyroid. So simply the error associated with acquiring the data makes it impossible to use this type of data to say that there's something wrong with the thyroid. I'll skip the blood pressure. There's a TEDx talk if you want to hear more about blood pressure, but these same algorithms that we've used to estimate tissue properties, we're developing a new technique to give a very rapid assessment of blood pressure. And the approach is quite, the algorithmic approach for how we analyze imagery as a function of force for just regular tissue is quite analogous to analyzing this imagery. This is an image of the carotid artery, acquired under from 3 to about 8 newtons of load. What you should see here is as we steadily increase the force, this is at the carotid, we're pressing harder and harder, the distance between the tissue, the skin, and the artery is getting smaller and smaller, while at the same time, the heart is beating, and so the artery is doing this. If we look at that, we see over that image sequence, on average, the small axis of the artery is getting smaller and smaller with this pulsation. So we're able to throw that data now into a deterministic model augmented with some learning, to extract not only the blood pressure, but the arterial stiffness, the stiffness of the tissue, and properties of the artery. We only currently can compare to a blood pressure cuff. This is a paper that came out in 2017 looking at 24 healthy volunteers, comparing our accuracy is about 10%. We already know that the blood pressure cuff is only good to about 10%, so we're not sure whether the error here or the difference here comes from our cuff or from the cuff that we're using or our technique. We're now going down the path with some funding from the NIH, the National Institutes of Health, to develop a sort of a bedside or vanity side device that you would just rapidly touch to your neck, to your leg, to your arm to get localized blood pressure. So I want to just talk about sort of the impact of pose and orientation. There are technologies that are deployed with GE and Philips that use electromagnetic trackers to track the position and orientation of the probe with respect to the room. And they use that for comparing preoperative data to during operative data, for example. Here, just this video is to highlight just a little bit of variation. This is on the kidneys, and this is on the thyroid. A little bit of variation of how you tilt the probe dramatically changes what you look at, or dramatically changes what you see. Certainly you can use 3D probes, so 2D arrays. There are some issues with those technologies as well in terms of cost. But this is now just sort of a series of examples showing how sensitive orientation and position of the probe on the body is. So there's a need to understand the orientation and position, to be able to track that orientation and position over time if we want to do serial measurements, or to combine now multiple orientations and positions in the same way that I've used multiple forces. So what we do, the new enhancement, is instead of adding a force sensor or a control system to the outside of the probe, we add cameras. As it turns out, your skin, the micro-reliefs and the texture on your skin, you know your fingerprints are unique. They can be used for analysis and biometrics. A patch of your skin, the micro-reliefs, the hair follicles, the texture, is locally unique. Your skin features, if you're looking at them, can be used in two ways. I can use now a camera mounted to an ultrasound probe as an optical mouse, in effect, to know the relative distances of how I'm moving along the body. But more importantly, it actually is a global encoding system. So I can use these skin features to tell the doctor, I want to get back to that same location and same position today, 6 months from today. So I'll accelerate sort of some of the results here. This is just showing some of the camera systems, what skin looks like up close and personal, tracking skin, doing the in-the-lab tests to demonstrate that the technology is working, making phantoms with tumors of known size and validating that we get what we expect, comparing arteries that we extract with what we're seeing in MR. The video of the process looks like this. We have an ultrasound probe with a camera attached to it, sliding along the body, getting the skin imagery, and using that to create a map of the patient, a map of the outside world, the world being the body, and then using that to in a dimensionally accurate way to construct large volumes of ultrasound data that allow me now to have a more natural view, the volume view inside the body. So the way that we want to make these technologies accessible is that we don't want to just deploy these with highly trained sonographers or highly trained doctors. We want to make it so that the general practitioner can use the technology. So in the context of adding cameras and sort of constructing ultrasound imagery that's mapped to the human body, we basically make a game for them. We create a map of the body with the camera, and we acquire that at time 1. At time 2, 6 months later, we have a virtual object, a pyramid in this case, that we attach to the body. And all we do is tell the doctor to play the game, to line up the 5 points of the pyramid, to get the skin data, jointly analyzed with the ultrasound data, but mostly the skin data, get it aligned to the same 5 points. You're at the same location, same position, snap the image, and now I have very accurate dimensional repeatability on where I've acquired an image, which makes it far easier to track things over time. The combination of the technologies is, well, we get volume data that's dimensionally aligned to the body. We get elastograms or volume imagery that is a function of force. And so these technologies in combination give us the tools to make a doctor better, but actually highly reduces the variability, increases the information that's in the ultrasound data, so that you can then turn it over to either a deterministic or a physiological based or a learned model to extract actual properties. So with that, I thank you for your time, and if we have time for maybe one or two questions, I'll take it, but if not, catch me afterwards. Thank you.
Well, thank you, Brian Anthony. This was impressive. I think we do have time for one or two questions. Are there any particularly urgent ones? Yeah, back there. Do we have somebody with a microphone?
I just can try to talk loud.
Right. Yeah, I'll repeat
Okay.
the question. Yeah, go ahead, I can hear you.
Yeah.
Thank you very much. I'm a researcher at the RCB for the health department, and we are actually making research in the field of data analysis and using ultrasound images to acquire bony landmarks for 3-dimensional data analysis. And we combine the motion capture system with the 3D, with the ultrasound probe to get 3-dimensional images of the ultrasound, and was asking myself how you were able to get 3-dimensional information from the ultrasounds just by using the skin as a reference. I can think of having 2 dimensions, but how did you manage to get it really in a 3-dimensional?
If you're familiar with SLAM, SLAM is simultaneous localization and mapping. It's the idea that if I take a single camera, so you're all really good at stereo. You have 2 cameras you see in the world, you get depth, you get 3D, you can catch a ball. If you take a single camera in a roughly static world and you move that camera around with enough information, and assuming a mostly static world, I can simultaneously map the world and know where the camera was. In our case, the world is the patient. We move the camera. So we don't, from a single image, we don't know enough. But from multiple images, as we've moved along the body, we're able to simultaneously construct a map of the world, the patient's body, and know where the camera was. And since we know how the camera is attached to the probe, that allows us, in a dimensionally accurate way, to stack together the ultrasound images.
Thank you.
Certainly.
All right, so thanks again, Mr. Anthony. Big round of applause. And yes, the speakers also get a little present, I've been told. I'm not sure what it is, but I'm sure it's something healthy.